Right now, there's a biological barrier protecting you from flesh-eating flies, and it's made entirely of sterile insects. This isn't natural. It's one of the most ambitious scientific interventions in history, requiring the weekly release of millions of radiation-sterilized flies across multiple countries. Imagine entire herds in Texas spared the gruesome fate that once killed 500,000 cattle in just one outbreak. This scientific intervention has never stopped since the 1950s. Over 100 million sterile flies still stream from the sky each week to hold back a lethal parasite. But here's what makes this even more incredible. It's the only thing preventing a catastrophic parasite invasion that would devastate livestock across entire continents. The invisible army above your head. Aircraft drop these sterile males across Panama, Costa Rica, Nicaragua, Honduras, Guatemala, and southern Mexico. Small boxes of sterile male flies pop open in flight, scattering thousands across a grid of fields the size of small countries. The coordination required is staggering. Multiple countries synchronizing their releases to create overlapping zones of sterile fly coverage. Weather conditions, wind patterns, and seasonal breeding cycles all factor into deployment schedules that must be maintained week after week, year after year. This invisible shield works because sterile males compete with wild males for mates, but when females choose the sterile males, because a female screwworm mates just once before dying, a single sterile mating spells doom for that entire genetic line. The sheer volume of sterile flies creates overwhelming odds against successful reproduction by wild populations. In areas where the program operates effectively, sterile males can outnumber wild males by ratios of 100 to 1 or higher. The female flies have no way to distinguish between fertile and sterile males, so they mate with whatever males they encounter first. Most people walking through these protected territories have no idea they're surrounded by millions of deliberately sterilized insects. The flies look identical to their wild counterparts. Same flight patterns, same mating behaviors. The only difference is their inability to produce viable offspring. This biological masquerade is essential to the program's success because the sterile flies must remain sexually competitive to effectively intercept wild females. If this protection disappeared overnight, the consequences would unfold rapidly. Wild screwworm populations would explode within months, with females laying hundreds of eggs in livestock wounds. The parasites would spread northward through Mexico into Texas, Arizona, New Mexico, and California. Ranchers would face the same devastating losses their predecessors experienced before the program began. Hospital emergency rooms would start seeing cases of human myiasis, screwworm larvae eating flesh in wounds that seem minor at first. The program essentially wages biological warfare using the enemy's own species against itself. Instead of trying to kill screwworms with pesticides or barriers, scientists learned to create an army of reproductive saboteurs. These sterile flies carry the same instincts and behaviors as their wild relatives, but their biological weapons programmed to end genetic lines rather than continue them. The strategy exploits the screwworm's mating behavior as a vulnerability rather than fighting their physical presence directly. This weaponization of sterility required decades of research to perfect. Scientists had to determine optimal radiation doses that would sterilize males without making them too weak or behaviorally abnormal to compete for mates. Too much radiation would kill the flies or make them unfit for reproduction. Too little would leave them partially fertile, potentially creating resistant populations. The precise calibration took years of experimentation and field testing to achieve. Modern mass rearing facilities now produce 100 million flies weekly using precisely calibrated diets and radiation. Advanced genetic screening ensures only the healthiest flies enter the sterilization process. Quality control systems test samples from each batch to verify sterility rates and meet operational standards. Current facilities can process millions of flies simultaneously through industrial-scale radiation chambers. The logistics of moving this many insects from breeding rooms to sterilization chambers to packaging systems requires assembly line efficiency. Specialized containers maintain optimal temperature and humidity during transport to aircraft. Loading systems transfer millions of flies into aircraft dispensers within tight time frames to maintain fly viability. 7 decades of continuous operation means this program has never experienced a complete shutdown. Even during political upheavals, natural disasters, and global pandemics, the sterile fly factories kept running. Aircraft kept flying their missions. The program survived changes in government, shifts in scientific leadership, and multiple reorganizations of participating agencies. This consistency is crucial because any significant gap in deployment would allow wild populations to reestablish themselves. The dedication required to maintain this schedule is extraordinary. Breeding facilities operate around the clock, with staff working shifts to monitor insect health, adjust feeding schedules, and maintain optimal environmental conditions. Pilots fly predetermined routes regardless of weather conditions that would ground normal aircraft. Ground crews load aircraft in pre-dawn darkness to ensure timely deployment. Laboratory technicians process quality control samples within hours of collection. Climate change is now forcing program expansion beyond its traditional boundaries. Rising temperatures are creating suitable screwworm habitat in regions previously protected by cold winters. Northern Mexico and southern Texas are seeing extended breeding seasons that require year-round rather than seasonal deployment. New territories in South America need protection as warming temperatures make previously marginal habitats suitable for screwworm establishment. The program's response to climate change requires additional aircraft, expanded breeding facilities, and larger staff. Scientists are developing predictive models to identify areas likely to need protection before screwworm populations become established. Early intervention requires deploying sterile flies into areas where wild populations don't yet exist, essentially creating preemptive biological barriers. Recent expansion into Libya and other parts of North Africa demonstrates how quickly the program can mobilize when screwworm populations appear in new territories. These emergency deployments require massive resource allocation and international coordination. Aircraft must be positioned, facilities must be established, and local staff must be trained within months rather than years. The program's success depends on maintaining sterile fly production at levels that consistently overwhelm wild populations. Any reduction in deployment numbers creates opportunities for wild flies to find mates and reproduce successfully. The mathematics are unforgiving. If sterile males don't maintain overwhelming numerical superiority, the biological shield begins to fail. Current deployment schedules account for seasonal variations in wild fly populations, adjusting release numbers based on breeding cycle predictions. Summer months typically require higher deployment rates as wild populations peak. Winter deployments maintain baseline protection during periods of reduced wild fly activity, where delays can create gaps in coverage that must be compensated by increased releases once conditions improve. The program represents one of the largest coordinated biological interventions in history. Multiple countries synchronize their activities to create continental-scale protection. Research facilities share data on fly genetics, breeding techniques, and deployment strategies. International agreements ensure continued cooperation regardless of political changes within participating nations. This protection requires constant vigilance because screwworm populations can reestablish quickly if sterile fly deployment falters. The program's success creates a dependency that can never be abandoned without risking catastrophic consequences. Every week's deployment is essential to maintaining the biological barrier that protects millions of animals and humans from flesh-eating parasites. The massive resources invested in this program reflect the enormous economic and humanitarian stakes involved. The alternative, allowing screwworm populations to reestablish, would cause suffering and economic losses that dwarf the program's operational costs. This invisible army of sterile flies represents humanity's commitment to protecting itself and its animals from one of nature's most destructive parasites. But what exactly makes this enemy so devastating that it requires such an extraordinary response? The answer lies in understanding the biological machine that nearly brought livestock farming to its knees. When paradise became hell. Cochleomia hominivrax operates as a biological machine designed for one purpose, converting living flesh into more screwworm flies. These metallic blue-green insects with bright orange eyes possess sensory capabilities that make them among nature's most efficient parasites. Their antennae can detect the chemical signature of an open wound from distances exceeding 2 miles. Blood proteins, tissue damage markers, and bacterial compounds from infected wounds create a scent trail that draws female screwworms like a beacon. Once they lock onto a scent, they can track it across vast territories, navigating through dense forests and open grasslands with remarkable precision. The female screwworm approaches a wound with surgical precision, evaluating the site for optimal egg placement. She prefers fresh wounds with exposed tissue and adequate moisture to support larval development. Using her specialized ovipositor, she deposits clusters of cream-colored eggs directly into the womb cavity. Each female can lay up to 400 eggs in a single session, and she will repeat this process multiple times throughout her 3-week lifespan. The eggs attach firmly to wound tissue, making removal nearly impossible without causing additional damage to the host animal. Within 24 hours, the eggs hatch into tiny larvae equipped with powerful mandibles designed for cutting through living tissue. These larvae immediately begin feeding on healthy flesh surrounding the wound, secreting enzymes that break down proteins and prevent blood clotting. The feeding process keeps wounds open and expanding, creating perfect conditions for additional egg laying by other females. Each larva grows rapidly, molding through 3 distinct stages before reaching maturity. The feeding larvae emit chemical signals that attract more female flies, turning a single small wound into a massive infestation site. The expanding wound becomes a festering crater as hundreds of larvae consume healthy tissue. The host animal experiences intense pain and becomes increasingly weak as the wound grows larger and more infected. Secondary bacterial infections often develop, adding another layer of suffering to the already severe tissue damage. In severe cases, the larvae consume so much tissue that vital organs become exposed. Animals can die from blood loss, infection, or organ failure within days of initial infestation. Even animals that survive often suffer permanent disfigurement and reduced mobility from the extensive tissue damage. When livestock farming expanded across the American Southwest in the late 1800s, ranchers unknowingly created ideal conditions for screwworm population explosions. Millions of cattle, sheep, and goats concentrated in relatively small areas provided an abundant supply of potential hosts. Barbed wire fencing caused frequent cuts and scrapes as animals moved through pastures. Branding, castration, and dehorning procedures created fresh wounds that attracted screwworms from miles away. Newborn animals were especially vulnerable, with their umbilical cords providing perfect entry points for egg-laying females. The dense livestock populations allowed screwworm numbers to multiply exponentially. A single infected animal could produce thousands of adult flies within weeks. These new flies would then spread throughout the area, seeking fresh wounds on other animals. The cycle repeated continuously, with screwworm populations doubling and redoubling until entire regions became infested. Ranchers reported losing 30 to 50% of their newborn calves to screwworm attacks. Adult animals suffered constant reinfection, making normal ranching operations nearly impossible. Economic losses from screwworm infestations reached catastrophic levels by the 1930s. The United States livestock industry was losing an estimated $20 million annually to screwworm damage, equivalent to hundreds of millions in today's currency. Texas alone reported losses exceeding $5 million per year. Ranchers faced impossible choices: abandon livestock operations entirely or watch their animals suffer horrible deaths. Some operations went bankrupt after losing entire herds to screwworm attacks. The parasite was essentially destroying the economic foundation of rural communities across the Southwest. Traditional pest control methods proved completely ineffective against screwworms. Pesticides designed for external parasites couldn't reach larvae buried deep inside wound tissue. Spraying animals with insecticides had no impact on eggs already deposited in wounds. Even if pesticides could kill adult flies, new ones would migrate in from surrounding areas within days. The larva's protected position inside living tissue made them virtually immune to chemical treatment. Topical wound treatments might kill surface larvae, but deeper infestations continued developing regardless. Ranchers attempted various desperate measures to protect their animals. They built elaborate screening systems around birthing areas, but flies could still penetrate most barriers. Some tried keeping animals in enclosed barns, but this was impractical for large herds and did prevent wounds from other causes. Others employed full-time workers to inspect animals daily and treat wounds immediately, but this approach was both expensive and ineffective. Even minor delays in wound treatment could allow screwworms to establish infestations that became impossible to control. The screwworm's life cycle made conventional control strategies worthless. Unlike other agricultural pests that could be controlled through habitat modification or breeding resistant crops, screwworms required only wounded animals to complete their development. Every animal injury, no matter how minor, became a potential breeding site. Preventing all animal injuries was impossible in normal ranching operations. The parasite's mobility allowed it to spread rapidly between farms and across vast distances, making localized control efforts futile. Veterinary treatments for screwworm infestations were primitive and often ineffective. Manually removing larvae from wounds was time-consuming and caused additional trauma to already suffering animals. Chemical treatments that killed larvae often damaged surrounding healthy tissue. Many infested animals die from the treatment itself rather than the original infestation. Even successful treatments didn't prevent reinfection, as new flies would target the healing wounds within days. The escalating crisis demanded solutions that went beyond traditional pest management approaches. Screwworm populations were expanding their range, moving into previously unaffected areas as livestock operations spread. The parasite's efficient reproduction and dispersal capabilities made it nearly impossible to contain using conventional methods. Each breeding cycle produced more flies capable of traveling greater distances to find new hosts. The problem was becoming continental in scope, threatening livestock operations throughout North America. Government agencies and agricultural organizations recognized that incremental improvements to existing control methods would never solve the screwworm problem. The parasite's biological advantages were too significant to overcome through minor adjustments to current practices. The situation required a fundamentally different approach that attacked the screwworm's reproductive cycle, rather than trying to kill individual flies or larvae. Scientists needed to find a way to turn the screwworm's own biology against itself. The realization that conventional weapons were powerless against screwworms forced researchers to consider radical alternatives. Chemical pesticides, physical barriers, biological predators, and habitat modification had all failed to provide meaningful control. The screwworm's protected larval stage and mobile adult stage made it essentially invulnerable to traditional attack methods. Only by disrupting the reproductive process itself could scientists hope to gain an advantage. But the solution they would propose sounded more like science fiction than serious research. The nuclear solution nobody believed. Edward Knipling and Raymond Bushland proposed bombarding millions of screwworm flies with gamma radiation to make them sterile, then releasing them into the wild to mate with wild females and produce no offspring. The entomology establishment dismissed this idea as fantasy. Leading scientists argued that radiation would either kill the flies outright or damage them so severely they couldn't compete with healthy wild males for mates. The concept violated everything researchers understood about pest control, which focused on killing insects rather than manipulating their reproduction. The scientific community's resistance was understandable given the era's limited knowledge of radiation biology. Most radiation research in the 1950s dealt with high doses that caused immediate death or severe mutations. The idea that precise radiation exposure could sterilize without killing seemed impossible to most researchers. Critics pointed out that insects exposed to enough radiation to sterilize them would lose their vigor, making them worthless as competitors for wild females. Others argued that even if sterilization worked, producing enough sterile flies to overwhelm wild populations would be logistically impossible. Knipling and Bushland discovered this approach might work because screwworm reproduction depends on a single mating event. Female screwworms mate only once during their 3-week lifespan, storing sperm to fertilize multiple egg batches. If a female mated with a sterile male, she would waste her entire reproductive potential. This biological constraint created a vulnerability that radiation could exploit. Male screwworms produce sperm continuously, making their reproductive cells more susceptible to radiation damage than other body systems. The key insight was that radiation affects rapidly dividing cells first, particularly sperm-producing cells in insect testes. Lower radiation doses could destroy reproductive capacity while leaving other cellular functions intact. The scientists theorized that sterile males would retain normal behavior, flight capability, and sexual competitiveness while becoming reproductively useless. This selective sterilization would turn the males into biological weapons that looked and acted like normal flies but carried reproductive death. Initial laboratory experiments focused on determining radiation doses that achieved complete sterilization without compromising male fitness. Researchers exposed male screwworms to various gamma radiation levels, then tested their mating behavior and sperm viability. Too little radiation left males partially fertile, which could lead to resistant populations. Too much radiation killed flies or made them too weak to mate successfully. The optimal dose required precise calibration measured in thousands of rads. Early tests revealed that males exposed to 7,500 rads of gamma radiation became completely sterile while maintaining normal lifespan and mating behavior. These sterile males competed successfully with normal males for female attention. Laboratory studies confirmed that females mated with sterile males produced no viable offspring. More importantly, the sterile males showed no behavioral changes that would make them less attractive to females. Their flight patterns, pheromone production, and courtship behaviors remained identical to fertile males. The first field test occurred on Sanibel Island, Florida, in 1951. Researchers released sterile male screwworms into a small, isolated population to test whether the technique would work in natural conditions. The island's limited size made it perfect for monitoring results without risking contamination of mainland populations. Within months, the wild screwworm population on Sanibel Island collapsed as females mated with sterile males and produced no offspring. The test proved that laboratory results translated to field conditions. Success on Sanibel Island led to larger trials on Curacao, a Caribbean island with established screwworm populations. The Curacao test required producing thousands of sterile flies weekly, forcing researchers to develop industrial-scale breeding methods. They built facilities that could rear screwworm larvae on artificial diets, manage adult fly colonies, and process males through radiation chambers. The logistics were staggering compared to laboratory-scale operations, requiring coordination between breeding specialists, radiation technicians, and field deployment teams. The Curacao experiment succeeded beyond expectations. Researchers released approximately 400 sterile males per square mile weekly for several months. Wild screwworm populations disappeared entirely within 6 months, with no evidence of surviving breeding pairs. The success demonstrated that sterile insect technique could eliminate established populations from large areas. However, the Curacao operation also revealed the massive infrastructure needed for continental-scale deployment. Scaling up to protect entire countries would require breeding facilities capable of producing millions of flies weekly. Each facility needed climate-controlled rearing rooms, artificial diet production systems, adult fly management areas, and industrial radiation chambers. The flies had to be reared on standardized diets that produce healthy, competitive males. Adult flies required specific temperature, humidity, and lighting conditions to maintain reproductive behavior. The radiation sterilization process needed precise timing to treat flies at optimal ages for maximum effectiveness. The breeding process began with maintaining colonies of adult flies in large cages supplied with protein-rich food sources. Females laid eggs on artificial substrates that simulated womb conditions. Workers collected eggs daily and transferred them to rearing containers filled with nutrient-rich media. Larvae developed through 3 stages before pupating in sterile sand. Adult flies emerged from pupae and required several days of feeding before reaching sexual maturity. Quality control systems tested random samples from each batch to verify complete sterilization. Researchers mated sample males with fertile females and monitored egg development to confirm no viable offspring resulted. Any batch showing partial fertility was rejected entirely. The testing process required maintaining laboratory colonies of fertile flies for comparison studies. Quality control data tracked sterilization effectiveness across different seasons and production conditions. International agreements established protocols for cross-border cooperation in sterile fly programs. Countries had to coordinate release schedules to prevent gaps in coverage along national boundaries. Standardized procedures ensured consistent quality across different production facilities. Research sharing agreements allowed improvements developed in one location to benefit all participating programs. The realization that success demanded unprecedented coordination between scientists, governments, and military-style logistics operations marked the beginning of biological warfare on a continental scale. This wasn't pest control anymore. It was a sustained campaign requiring industrial production capacity and permanent commitment to maintaining operations indefinitely. The scale of infrastructure needed to wage this war would dwarf anything previously attempted in biological control, creating something unprecedented in biological control. The Copen facility in Guatemala stretches across 30 acres of industrial buildings that operate like a biological weapons factory disguised as a research center. Inside these climate-controlled structures, workers tend to millions of screwworm flies in various stages of development. The facility produces approximately 40 million sterile males weekly, requiring a production pipeline that never stops. Each building serves a specific function in the assembly line of death egg collection rooms, larval rearing chambers, pupation areas, adult fly colonies, and radiation sterilization facilities. Modern breeding operations bear no resemblance to the primitive meat-based systems used in early experiments. The original facilities fed screwworm larvae on chunks of rotting meat, creating horrific working conditions and inconsistent nutrition that produce weak flies. Today's operations use precisely formulated artificial diets that provide optimal nutrition for rapid development and maximum adult vigor. The synthetic diet contains exact ratios of proteins, carbohydrates, vitamins, and minerals that produce flies superior to wild populations in size and competitive ability. The production process begins with maintaining breeding colonies of adult flies in massive screened enclosures. These colonies require constant monitoring to ensure proper sex ratios, adequate nutrition, and optimal environmental conditions. Workers collect eggs daily from artificial substrates designed to simulate womb conditions. Each egg batch is tracked through the entire production cycle with barcode systems that monitor development timing, diet consumption, and emergence rates. Quality control begins at the egg stage with microscopic examination to identify any abnormalities that might affect adult fly performance. Larval rearing occurs in temperature-controlled rooms filled with thousands of containers holding developing flies. The containers are arranged in precise grids that allow automated feeding systems to deliver measured amounts of artificial diet at programmed intervals. Each container holds exactly 1,000 larvae to prevent overcrowding that would reduce individual fly size. Temperature controllers maintain conditions within 1 degree of optimal development ranges. Humidity systems ensure proper moisture levels that prevent diet spoilage while supporting healthy larval growth. The transformation from larvae to adult flies requires 3 weeks under carefully controlled conditions. Workers monitor development progress hourly, adjusting environmental factors to maintain synchronous development across millions of individuals. Pupation occurs in sterile sand beds that simulate natural conditions while preventing contamination. Adult flies emerge in the holding areas where they feed on sugar solutions and protein supplements designed to maximize their reproductive vigor. Male separation happens through sophisticated mechanical systems that sort flies based on size differences between sexes. The sorting process must handle thousands of flies per hour without damaging their delicate bodies or reducing their competitive ability. Workers use temperature manipulation to slow fly movement during sorting, then employ specialized equipment to separate males based on anatomical features. Only the largest, most vigorous males advance their radiation treatment. The radiation chambers represent the most critical component of the entire operation. These lead-line rooms contain cobalt-60 sources that emit precisely controlled gamma radiation doses. Males are placed in specially designed containers that ensure uniform radiation exposure across thousands of flies simultaneously. The radiation dose must be exact 7,500 rads, delivered over a specific time period that sterilizes without killing. Dosimetry equipment monitors radiation levels continuously to ensure consistent treatment. Quality control systems test random samples from each batch to verify complete sterilization while maintaining competitive ability. Laboratory technicians mate sample males with fertile females and monitor egg development under microscopes to confirm zero fertility. Behavioral tests evaluate whether sterile males display normal flight patterns, feeding behaviors, and mating responses. Any batch showing reduced vigor or incomplete sterilization is destroyed entirely, regardless of production cost. The scale of operations requires sophisticated logistics coordination. Raw materials for artificial diets arrive by the ton, requiring storage facilities and mixing equipment that can produce thousands of gallons of larval food daily. Adult flies consume massive quantities of sugar solutions and protein supplements that must be prepared fresh continuously. The facility generates enormous amounts of waste material from spent diet, dead flies, and contaminated equipment that requires specialized disposal methods. Packaging systems prepare sterile males for transport in containers designed to maintain optimal survival during shipment. Each container holds exactly 2,000 flies with ventilation systems that provide adequate airflow without allowing escape. Temperature control during packaging prevents thermal stress that can reduce fly longevity. The containers are labeled with production dates, batch numbers, and quality control data that track each group of flies through deployment. The workforce operates in shifts that maintain production 24 hours daily, 7 days weekly. Different teams specialize in egg collection, larval care, adult fly management, radiation treatment, quality control, and packaging operations. Each worker requires extensive training in sterile technique, insect handling, and safety protocols. The facility employs entomologists, radiation technicians, quality control specialists, and maintenance crews who keep the complex systems functioning without interruption. The precision required in every operation step cannot be overstated. Slight variations in diet formulation affect adult fly size and vigor. Temperature fluctuations during development alter emergence timing and synchronization. Radiation dose variations can leave flies partially fertile or reduce their competitive ability. Packaging delays can increase mortality during transport. Each component must function perfectly to maintain the massive output required for continental protection. These facilities function as weapons factories in humanity's biological war against screwworm flies. Every sterile male produced represents a potential reproductive dead end for wild populations. The industrial scale of production creates an overwhelming numerical advantage that makes wild fly reproduction nearly impossible. But producing millions of sterile flies means nothing without a delivery system capable of placing them exactly where they need to be across thousands of square miles of terrain. Precision deployment warfare. Aircraft operations transform these biological weapons into precisely targeted strikes against wild screwworm populations. Twin-engine Cessna aircraft take off from airstrips across Central America every morning at calculated times, their cargo holds filled with millions of sterile flies ready for deployment. These flights follow GPS coordinates programmed down to the meter, creating invisible grid patterns that ensure complete coverage without gaps or overlaps. The aircraft fly at altitudes between 500 and 1,000 feet, low enough to deliver flies accurately but high enough to avoid terrain obstacles. The release mechanisms mounted beneath these aircraft represent decades of engineering refinement. Specialized containers hold exactly 2,000 sterile flies each, with pneumatic systems that split the containers open at predetermined GPS coordinates. The timing must be precise because flies released too early or late create gaps in coverage that wild flies can exploit. Computer systems calculate optimal release points based on real-time wind data, ensuring flies land within designated zones rather than drifting into areas already covered. Ground control coordinates multiple aircraft simultaneously, tracking each plane's position and release schedule to satellite monitoring systems. Controllers adjust flight paths in real time based on weather conditions, air traffic, and ground obstacles. The coordination resembles air traffic control for a major airport, except these flights carry biological weapons instead of passengers. Radio communications between aircraft and ground control use coded terminology to maintain operational security while coordinating complex deployment patterns. Flight patterns create overlapping zones of sterile fly coverage that prevent wild flies from finding gaps in the barrier. Each aircraft follows predetermined routes that place sterile flies at specific intervals across the landscape. The space in between release points is calculated to ensure sterile flies can disperse naturally to achieve optimal population density ratios. Too much spacing creates gaps where wild flies can establish breeding populations. Too little spacing wastes resources by concentrating sterile flies in areas already adequately covered. Weather conditions determine whether flights can proceed or must be delayed. Wind speeds exceeding 15 miles per hour scatter flies too widely to maintain effective coverage. Rain kills flies during deployment or immediately after landing, making releases during precipitation completely wasteful. Temperature extremes above 95 degrees or below 60 degrees reduce fly survival rates and competitive ability. Cloud cover affects pilot visibility and GPS accuracy, potentially compromising release precision. Seasonal variations require adjusting deployment schedules to match wildfly breeding cycles. Spring deployments begin early to establish sterile fly populations before wild flies emerge from winter dormancy. Summer operations increase release frequencies to match peak wild fly reproduction periods. Fall deployments gradually reduce as wild populations decline with cooler temperatures. Winter operations maintain baseline coverage in tropical regions where wild flies remain active year-round. Terrain features complicate deployment strategies because flies must land in areas where they can survive and encounter wildflies. Mountainous regions create wind patterns that scatter flies unpredictably, requiring adjusted release altitudes and timing. Dense forest canopies prevent flies from reaching ground level where animals are located. Open water areas provide no suitable habitat for fly survival. Urban areas present navigation hazards and potential public concerns about releasing insects. The specialized aircraft used in current operations are modified civilian planes equipped with GPS navigation systems accurate to within 3 meters. Multiple backup navigation systems ensure continued operation if primary GPS signals fail. Communication equipment allows constant contact with ground control and other aircraft in the deployment area. Emergency procedures enable pilots to abort missions if equipment failures threaten release accuracy or flight safety. Loading operations begin before dawn at airstrips near breeding facilities. Ground crews transfer containers of sterile flies from climate-controlled storage into aircraft cargo holds within tight time frames to maintain fly viability. Each container is checked for proper sealing and labeled with GPS coordinates for specific release points. Loading procedures follow strict protocols to prevent accidental release during ground operations or flight preparation. Real-time monitoring systems track deployment effectiveness through multiple data sources. Satellite imagery provides visual confirmation of aircraft positions and release patterns. Ground-based monitoring stations collect data on sterile fly survival rates and distribution patterns. Field teams collect samples to verify sterile fly presence and competitive ability in target areas. Weather monitoring systems track environmental conditions that affect fly survival and behavior. Computer modeling systems predict optimal deployment patterns based on historical data, current weather conditions, and wild fly population estimates. These models calculate required sterile fly densities to achieve effective suppression ratios. Deployment schedules are adjusted based on modeling predictions to maximize program effectiveness while minimizing operational costs. The models account for seasonal variations, weather patterns, and terrain features that affect fly distribution and survival. Aircraft from different countries coordinate their flight schedules to prevent gaps in coverage along border regions. Standardized release procedures ensure consistent fly distribution regardless of which country's aircraft performs the deployment. Emergency protocols allow aircraft from one country to cover areas normally handled by neighboring nations if equipment failures or weather delays disrupt normal operations. The precision required in deployment operations leaves no room for error. Flight crews must maintain exact altitudes and speeds to ensure accurate release timing. Navigation systems must function perfectly to place flies at predetermined coordinates. Release mechanisms must operate consistently to deliver flies at specified intervals. Weather monitoring must provide accurate predictions to determine optimal deployment conditions. Each deployment flight represents a calculated strike in an ongoing biological war against screwworm flies. The sterile flies released during these missions are weapons designed to seek out and neutralize wild flies through reproductive sabotage. The precision of deployment operations determines whether these biological weapons reach their targets effectively. Every flight must succeed because gaps in coverage allow wild flies to reestablish populations that threaten the entire protection system. But the true measure of this massive operation success isn't counted in flies deployed or flights completed. The economic shield worth billions. Success gets measured in dollars saved and disasters prevented. The Sterile Fly program prevents approximately $3.2 billion in annual livestock losses across participating countries, making it one of the most cost-effective biological interventions in history. This figure represents direct savings from avoided animal deaths, reduced veterinary costs, and prevented productivity losses in cattle, sheep, goat, and pig operations. The United States alone saves an estimated $1.8 billion annually, while Mexico prevents $800 million in losses, and Central American countries collectively avoid $600 million in agricultural damage. Calculations extend far beyond simple livestock mortality figures. Screwworm infestations reduce milk production in dairy cows by 30 to 50% during active infections. Beef cattle lose significant weight as their energy goes toward fighting infections rather than muscle development. Breeding animals suffer reduced fertility rates, with some females becoming permanently sterile from severe infestations. The program prevents all these productivity losses, maintaining agricultural output at levels that would be impossible with active screwworm populations. If the sterile fly program failed for just 6 months, economic modeling predicts catastrophic losses would begin within 60 days. Wild screwworm populations would explode exponentially, with females laying eggs in livestock wombs throughout the previously protected areas. Texas ranchers would face immediate losses exceeding $200 million monthly as newborn calves became primary targets for screwworm attacks. California's dairy industry would experience production drops worth $150 million monthly from infected cows. Arizona and New Mexico cattle operations would lose an estimated $100 million monthly from animal deaths and reduced productivity. The program's annual operating costs total approximately $45 million across all participating countries, creating a cost-benefit ratio of more than 70 to 1. This includes breeding facility operations, aircraft deployment, personnel salaries, research and development, and international coordination expenses. The United States contributes $25 million annually, Mexico provides $12 million, and Central American countries collectively fund $8 million. These operational costs are insignificant compared to the economic devastation that would result from program failure. Private sector investments amplify the program's economic impact through industries that depend on livestock production. Feed manufacturers, veterinary suppliers, meat processing plants, leather producers, and dairy operations all benefit from stable livestock populations. The program supports approximately 180,000 jobs in livestock-dependent industries across participating countries. These employment figures include ranchers, veterinarians, feed mill workers, meat packers, and transportation personnel whose livelihoods depend on healthy animal populations. Banking and insurance sectors rely on the program's continued success to maintain agricultural lending portfolios. Livestock serves as collateral for billions in agricultural loans, with banks requiring healthy herds to secure financing. Insurance companies base their livestock coverage rates on the assumption that screwworm infestations will not occur. Program failure would trigger massive loan defaults and insurance claims that could destabilize rural banking systems across multiple countries. Export markets depend on screwworm-free certification for international trade agreements. Countries with active screwworm populations face import restrictions and quarantine requirements that eliminate their ability to export livestock products. The United States exports $2.4 billion in beef annually, with much of this trade dependent on maintaining screwworm-free status. Mexico's $800 million beef export industry would collapse immediately if screwworm populations reestablished. Trade agreements specifically reference screwworm control as a requirement for continued market access. The program represents one of the most successful government interventions in agricultural history, with documented success spanning 7 decades. No other biological control program has achieved comparable scale, longevity, or economic impact. The eradication of screwworms from the United States stands as a rare example of permanently eliminating an agricultural pest through sustained scientific intervention. The program's success model has been replicated for other pest species, creating additional economic benefits beyond screwworm control. Regional economic development depends on agricultural stability that the program provides. Rural communities throughout the protected areas base their economies on livestock production that would be impossible without screwworm control. Small towns support feed stores, veterinary clinics, equipment dealers, and other businesses that serve ranching operations. The program's economic impact ripples through entire regional economies, supporting businesses far removed from direct livestock production. The program's economic shield protects agricultural assets worth an estimated $400 billion across all participating countries. This includes livestock populations, related infrastructure, processing facilities, and supporting industries. The protection extends beyond current agricultural assets to include future productivity and expansion potential. Without this shield, entire regions would be unsuitable for livestock production, eliminating billions in potential economic development. This invisible economic protection operates continuously, preventing losses that would cascade through multiple economic sectors. The program functions as agricultural insurance that costs pennies compared to the economic devastation it prevents. Every sterile fly release represents a small investment that protects thousands of dollars in agricultural assets. The mathematics are overwhelming. This biological shield protects trillions in economic value through sustained deployment of millions of sterile insects. But the foundation supporting this economic fortress is shifting beneath its feet. Environmental changes are forcing the program to expand into territories once considered permanently safe, requiring new investments and strategies that challenge the established operational model. Climate change reshapes the battlefield. Rising temperatures are rewriting the rules of biological warfare. Temperature data from 2025 reveals a disturbing trend that threatens to unravel decades of screwworm control success. Average winter temperatures across northern Mexico have increased by 3.2 degrees Fahrenheit since 2010, creating year-round survival conditions for screwworm flies in areas previously protected by seasonal cold snaps. What used to be a natural barrier that killed screwworm populations during winter months no longer exists in many regions. The flies now survive through winter in areas where they once died off completely, creating permanent breeding populations that require continuous sterile fly deployment. Southern Texas is experiencing the most dramatic shift in screwworm risk profiles. Towns like Brownsville, McAllen, and Laredo now record fewer than 10 days annually with temperatures below 40 degrees Fahrenheit. These brief cold periods are insufficient to eliminate screwworm populations that can survive short temperature drops by seeking shelter in protected areas. The traditional seasonal deployment schedule that protected these areas during summer months while allowing winter respite no longer works. Year-round deployment operations now cover 15,000 square miles of Texas territory that previously required only seasonal protection. The expansion of suitable screwworm habitat is forcing program managers to deploy sterile flies into areas where wild populations don't yet exist. This preemptive approach prevents screwworm establishment rather than waiting for infestations to develop. Northern Mexico regions around Monterrey and Saltillo now receive monthly sterile fly releases based on climate projections showing probable screwworm survival within 5 years. The strategy requires producing additional millions of sterile flies for areas that may not need protection for several years. Predictive modeling systems analyze temperature, humidity, and precipitation data to identify territories likely to become screwworm suitable within the next decade. These models process satellite weather data, ground station measurements, and historical climate patterns to generate risk maps updated quarterly. The models predict that screwworm's suitable habitat would expand northward by approximately 50 miles within the next 10 years. Areas of Colorado, New Mexico, and Arizona that have never supported screwworm populations may require protection by 2030. Changing precipitation patterns complicate deployment strategies by creating seasonal variations in screwworm survival rates. Increased rainfall in desert regions provides additional moisture that supports screwworm development in areas previously too dry for reproduction. Extended drought periods in traditionally humid regions can eliminate screwworm populations temporarily, but also reduce the effectiveness of sterile fly deployment by concentrating flies in smaller areas. Weather monitoring systems now track moisture levels at thousands of locations to adjust deployment schedules based on changing survival conditions. Hurricane seasons create additional challenges for maintaining continuous deployment schedules. Tropical storms disrupt aircraft operations and destroy ground-based facilities, creating gaps in sterile fly coverage that wild populations can exploit. The 2025 hurricane season forced suspension of deployment operations across Guatemala and Honduras for 3 weeks, requiring emergency increases in sterile fly production once weather conditions improved. Storm tracking systems now provide 10-day forecasts that allow preemptive adjustments to deployment schedules and sterile fly production rates. Temperature extremes beyond normal ranges affect sterile fly survival after deployment. Heat waves exceeding 15 degrees Fahrenheit reduce sterile fly longevity and competitive ability, requiring increased deployment frequencies during extreme weather events. Unexpected cold snaps can kill newly deployed sterile flies before they encounter wild populations, wasting production resources and creating temporary gaps in coverage. Weather monitoring systems track temperature extremes to optimize deployment timing and avoid wasting sterile flies in unsurvivable conditions. The program's adaptation strategies include developing heat-resistant sterile fly strains that maintain competitive ability at higher temperatures. Research facilities are selecting breeding lines that produce flies capable of surviving in the expanded temperature ranges created by climate change. These genetic improvements require extensive testing to ensure heat tolerance doesn't reduce mating competitiveness or longevity. Field trials compare heat-resistant strains with standard flies to verify equivalent performance in wild population suppression. Deployment aircraft now carry additional navigation and weather monitoring equipment to handle increased weather variability. Pilots receive enhanced training in extreme weather flying techniques and emergency procedures for sudden weather changes. Flight routes include alternate landing sites and fuel reserves to accommodate weather delays and course changes. Communication systems provide real-time weather updates to aircraft crews and ground control during deployment operations. The economic impact of climate-driven program expansion includes additional breeding facilities, aircraft, and personnel required for extended operations. Annual program costs have increased by $12 million since 2020 due to climate-related expansion needs. New breeding facilities in northern Mexico and southern Texas produce an additional 30 million sterile flies weekly to cover expanded deployment areas. Additional aircraft and flight crews provide deployment capacity for the larger geographic coverage required by changing climate conditions. Emergency response capabilities require rapid deployment capacity when climate conditions suddenly create new screwworm suitable areas. Mobile breeding units can establish temporary production capacity within weeks of identifying new risk areas. Emergency protocols activate additional aircraft and personnel to provide immediate deployment capacity for newly threatened regions. Stockpiled sterile flies provide temporary protection while permanent facilities are established in new coverage areas. The program's evolution reflects humanity's adaptation to climate change through technological innovation and international cooperation. The biological war against screwworm flies now requires constant adaptation to changing environmental conditions that expand the battlefield continuously. Climate change transforms this from a stable protection program into a dynamic response system that must evolve continuously to maintain effectiveness. Managing this expanding war demands precision that exceeds human capabilities, requiring systems that can process vast amounts of data and make split-second decisions across multiple variables simultaneously. Advanced computer systems now orchestrate every phase of this biological warfare operation with precision that surpasses human oversight. Artificial intelligence monitors individual larvae development while calculating optimal radiation doses for maximum sterilization effectiveness. Machine learning algorithms process millions of data points from breeding facilities, tracking temperature fluctuations, humidity levels, diet consumption rates, and larval growth patterns. These systems detect problems before human operators notice them, adjusting environmental controls automatically to maintain optimal conditions. The AI learns from historical data to predict equipment failures, schedule maintenance during low-impact periods, and optimize resource allocation across multiple production cycles. Computer vision systems examine individual flies during sorting, identifying males with surgical precision while rejecting females and substandard specimens. High-resolution cameras capture images of thousands of flies per minute, with pattern recognition software analyzing wing patterns, body size, and anatomical features that distinguish healthy males from inferior specimens. The system rejects flies showing signs of disease, developmental abnormalities, or reduced vigor that could compromise their competitive ability in the field. This automated inspection eliminates human error while processing volumes impossible for manual sorting operations. Genetic monitoring systems track DNA profiles of sterile fly populations to ensure they remain genetically competitive with wild populations. Laboratory technicians extract DNA samples from production flies and wild flies collected from field sites, comparing genetic markers that indicate fitness, disease resistance, and mating success. The genetic data reveals whether sterile flies maintain the same genetic diversity as wild populations or if inbreeding in production facilities is reducing their competitive ability. Any genetic drift that could compromise program effectiveness triggers immediate adjustments to breeding colony management. Radiation-sterilization systems use computer-controlled dosimetry to deliver precise gamma-ray exposure to millions of flies simultaneously. Sensors monitor radiation levels throughout sterilization chambers, ensuring uniform exposure across all flies while preventing overdoses that could kill or weaken them. The automated systems adjust exposure timing based on fly age, size, and batch characteristics to optimize sterilization while maintaining competitive vigor. Quality control systems test random samples from each batch, with results fed back into computer systems to refine radiation parameters for future batches. Data analytics platforms process information from breeding facilities, deployment operations, and field monitoring systems to identify patterns that predict program success or failure. The analytics identify optimal production timing, deployment schedules, and environmental conditions that maximize wild population suppression. Historical data spanning decades enables predictive modeling that anticipates problems before they affect program effectiveness. Satellite monitoring systems track deployment aircraft in real time, verifying that sterile flies are released at correct coordinates and timing intervals. GPS tracking data combines with weather satellite information to confirm optimal deployment conditions and document coverage patterns. The satellite systems monitor vegetation changes, livestock distribution, and environmental conditions that affect screwworm habitat suitability. This orbital surveillance provides early warning of areas where wild screwworm populations might establish before ground-based monitoring detects them. Automated breeding systems maintain optimal environmental conditions across thousands of rearing containers without human intervention. Sensors monitor temperature, humidity, air circulation, and lighting conditions continuously, with computer systems adjusting environmental controls based on program parameters. Automated feeding systems deliver precise amounts of artificial diet at scheduled intervals, with consumption rates monitored to detect health problems or developmental issues. The systems maintain detailed records of every environmental variable, creating databases that optimize conditions for maximum fly production. Laboratory information management systems track millions of flies through the entire production pipeline, from egg collection through deployment operations. Barcode systems identify individual batches, recording development timing, treatment protocols, and quality control results. The database systems link production data with field performance metrics, revealing which production variables correlate with effective wild population suppression. This comprehensive tracking enables rapid identification of problems and immediate correction of production processes. Advanced aircraft navigation systems guide deployment planes along precise flight paths using GPS coordinates accurate to within 3 meters. Automated release systems trigger container opening at exact geographic coordinates, ensuring consistent fly distribution across target areas. Weather monitoring systems provide real-time updates on wind speed, temperature, and precipitation that affect deployment effectiveness. The navigation systems compensate for wind drift and atmospheric conditions to place flies accurately within designated zones. Quality control systems test thousands of flies daily to verify sterilization effectiveness and competitive performance. Automated testing equipment mates sample flies with fertile females, then monitors egg development under computer-controlled microscopes. Image analysis software counts fertile versus infertile eggs, calculating sterilization rates with precision impossible through manual counting. Behavioral testing systems evaluate fly mating responses, flight performance, and longevity to ensure sterile flies remain competitive with wild populations. Environmental monitoring networks collect data from thousands of locations across protected territories, tracking conditions that affect screwworm survival and reproduction. Automated weather stations record temperature, humidity, precipitation, and wind patterns that influence deployment timing and fly survival rates. Soil moisture sensors and vegetation monitors provide data on habitat conditions that support screwworm development. This environmental surveillance guides deployment decisions and identifies areas requiring modified protection strategies. Emergency response systems activate automatically when monitoring systems detect potential program failures or wild screwworm outbreaks. Alert protocols notify key personnel immediately when monitoring systems identify unusual patterns or equipment failures. Automated response systems can increase production rates, adjust deployment schedules, or activate backup facilities without waiting for human decision-making. These rapid response capabilities prevent small problems from escalating into major program disruptions. The integration of these technological systems creates a comprehensive biological warfare platform that operates with precision impossible through manual management. Every aspect of sterile fly production, deployment, and monitoring relies on computer systems that process information faster and more accurately than human operators. This technological foundation enables a massive scale and consistent effectiveness. But even with this technological superiority, the program's effectiveness varies dramatically across different regions and environmental conditions. Success rates across the globe. The United States and Mexico report screwworm suppression rates exceeding 99.8% across their protected territories, representing the gold standard for sterile fly program effectiveness. These regions benefit from decades of operational experience, sophisticated breeding facilities, and well-established deployment networks that maintain consistent coverage. The success stems from ideal geographic conditions, including predictable weather patterns, accessible terrain for aircraft operations, and cooperative livestock industries that report suspected cases immediately. Border regions between the two countries show particularly strong results because overlapping deployment schedules create redundant protection that prevents any gaps in coverage. Central American countries demonstrate more variable success rates that reflect a challenging operational environment in tropical regions. Costa Rica and Panama maintain suppression rates above 95% through intensive deployment schedules and rapid response protocols. However, Guatemala and Honduras struggle with success rates between 85 and 92% due to difficult terrain, unpredictable weather patterns, and limited infrastructure for monitoring operations. The dense jungle canopy in these regions prevents effective aircraft deployment in many areas, while mountainous terrain creates wind patterns that scatter flies beyond target zones. The Libya operation launched in 2024 provides dramatic evidence of how geographic factors influence program effectiveness. The arid desert environment initially seemed ideal for sterile flight deployment because clear skies and minimal vegetation allowed precise aircraft navigation and flight placement. However, extreme temperature fluctuations between day and night reduced sterile fly survival rates below operational thresholds. Daytime temperatures exceeding 115 degrees Fahrenheit killed newly deployed flies before they could encounter wild populations, while nighttime temperatures dropping below 50 degrees further reduced fly longevity and reproductive behavior. Desert operations require completely different deployment strategies compared to tropical or temperate regions. Aircraft must deploy flies during narrow temperature windows in early morning or late evening when conditions support fly survival. The timing creates logistical challenges because flies must be transported from breeding facilities to deployment sites within precise time frames that account for temperature variations. Sandstorms and dust clouds create additional deployment hazards that ground aircraft and reduce visibility for precise navigation. Island territories demonstrate unique challenges that affect program success rates differently than continental operations. The Caribbean islands show excellent suppression rates above 98% because their isolated geography prevents wildfly migration from unprotected areas. However, the limited land area creates deployment challenges because aircraft must achieve precise coverage without wasting flies over water or unsuitable habitat. Island operations require smaller aircraft with enhanced navigation systems and modified release mechanisms that account for coastal wind patterns and limited landing strip availability. Mountainous regions consistently show the lowest program success rates due to complex terrain that disrupts deployment patterns and creates isolated pockets where wildflies can establish breeding populations. The Andes Mountains in South America create wind shear patterns that scatter deployed flies across vast areas, reducing effective coverage density. Aircraft must navigate between peaks and through valleys while maintaining precise release timing, creating safety hazards that limit deployment frequency. Ground-based monitoring becomes extremely difficult in remote mountain areas where access requires helicopters or multi-day hiking expeditions. Success measurement systems vary significantly between regions based on available infrastructure and monitoring capabilities. Advanced regions like the United States employ extensive networks of automated monitoring stations, satellite tracking systems, and laboratory analysis programs that provide real-time data on program effectiveness. These systems detect wildfly populations within days of establishment and trigger immediate response protocols. Less developed regions rely on manual monitoring through field teams that collect samples monthly and process results in basic laboratory facilities that may take weeks to provide definitive identification. Recent expansion efforts into Northern Africa and Southern Europe provide insights into how quickly programs can establish effectiveness in new territories. The initial deployment phases typically achieve 60 to 70% suppression rates within 6 months, improving to 85% within 1 year as operational procedures adapt to local conditions. However, these early success rates depend heavily on rapid response to initial wildfly detections and intensive deployment schedules that may not be sustainable long-term. The learning curve for new territories requires 18 to 24 months to achieve stable suppression rates comparable to established operations. Climate conditions create the most significant variable affecting program success across different regions. Areas with stable temperatures and predictable seasonal patterns consistently outperform regions with extreme weather variability. Sudden temperature changes, unexpected rainfall, or prolonged drought periods can reduce program effectiveness by 10 to 15% until deployment schedules adjust to new conditions. Hurricane-prone regions face periodic disruptions that create temporary gaps in coverage, requiring emergency response protocols and increased production capacity to compensate for weather-related delays. Emergency response capabilities significantly influence overall program success rates because rapid intervention prevents small wild fly populations from establishing permanent breeding colonies. Regions with helicopter access and mobile laboratory units can respond to detections within 24 hours, often preventing population establishment entirely. Areas requiring days or weeks to mobilize response teams frequently face more extensive infestations that require months of intensive treatment to eliminate. The difference between immediate and delayed response often determines whether a detection becomes a minor incident or a major operational challenge. International cooperation becomes essential for maintaining program effectiveness across national boundaries because wildflies do not recognize political borders. Regions with strong international coordination show higher success rates than areas where individual countries operate independently. Standardized procedures, shared monitoring data, and coordinated deployment schedules create seamless protection that prevents wildflies from exploiting gaps between national programs. Even the most successful programs remain vulnerable to sudden failures that can unravel decades of protection within weeks. The sophisticated monitoring systems that track these success rates also serve as early warning networks for the inevitable moments when the biological shield develops cracks. When the shield fails. Detection systems caught the first crack in September 2024 when automated sensors at a Guatemala monitoring station identified wild screwworm flies in territory protected for over 40 years. DNA analysis confirmed fertile females carrying viable eggs near the Mexican border. Within hours, emergency protocols activated across 3 countries as scientists realized wild flies had penetrated the sterile fly barrier. The breach occurred during Hurricane Helga, which disrupted deployment operations for 6 consecutive days across Guatemala and southern Mexico. Aircraft couldn't fly in storm conditions, creating gaps in sterile fly coverage that allowed wild populations to establish breeding colonies. The hurricane's aftermath revealed how quickly screwworm populations explode when protection fails. A single breeding pair detected Monday had become an estimated 2,000 wild flies by Thursday, with females laying eggs in wombs across multiple ranches. Emergency response teams mobilized within 12 hours, deploying mobile laboratories and helicopter monitoring units to assess the outbreak extent. The response required coordinating personnel from the United States, Mexico, and Guatemala, with aircraft redirected from routine operations to focus on the outbreak zone. Mobile breeding units positioned at temporary facilities produced additional sterile flies specifically for the emergency area. The resource mobilization for a single outbreak demonstrated the massive infrastructure required to maintain continental biological protection. Advanced monitoring systems detected this breach faster than any previous outbreak in program history. DNA analysis identified wild flies within 24 hours of collection, compared to week-long delays common in earlier decades. Satellite tracking guided field teams to specific locations where wildflies were most likely found based on livestock distribution and wound probability calculations. Automated alerts notified key personnel immediately when laboratory results confirmed wildfly presence, eliminating communication delays that had compromised previous emergency responses. The race against time became critical as wild screwworm populations double every 10 days under optimal conditions. Field teams worked around the clock locating and treating wounded animals in the outbreak zone, removing potential breeding sites before females could lay eggs. Aircraft flew deployment missions every 6 hours instead of the normal weekly schedule, flooding the area with sterile males to overwhelm remaining wildflies. The intensive deployment required producing 50 million additional sterile flies within 2 weeks, straining production facilities to maximum capacity. Resource mobilization included 12 aircraft, 200 personnel, and mobile laboratory units processing thousands of samples daily. Breeding facilities in Mexico and the United States increased production by 30% to provide additional sterile flies for emergency deployment. Helicopter teams conducted aerial surveillance of livestock herds, identifying animals with fresh wounds that might attract wildflies. Ground teams inspected over 15,000 animals across 200 ranches, treating wounds and collecting samples for laboratory analysis. The Guatemala outbreak revealed the fragility of even the most successful biological control programs. 7 decades of continuous protection had created false security that the sterile fly barrier was impenetrable. The hurricane demonstrated how natural disasters could disrupt deployment schedules and create vulnerabilities wild flies could exploit. A single week without adequate sterile fly coverage nearly compromised the entire regional protection system, threatening to restart screwworm establishment throughout Central America. Emergency response costs exceeded $8 million during the 3-month eradication effort, demonstrating enormous expense required to eliminate wild populations once established. Financial impact included increased sterile fly production, additional aircraft operations, overtime personnel costs, and mobile laboratory deployments. Emergency funding came from contingency reserves maintained by participating countries specifically for outbreak responses. The costs highlighted the economic wisdom of maintaining continuous protection rather than allowing periodic outbreaks. Similar breaches occurred near the Texas-Mexico border, when equipment failures at a key breeding facility reduced sterile fly production by 40% for 2 weeks. The production shortage created deployment coverage, gaps that allowed wild flies to establish breeding colonies in 3 separate locations. Each outbreak required individual emergency responses that collectively involved 18 aircraft and 300 personnel. Multiple simultaneous outbreaks strained emergency response capabilities and demonstrated system vulnerability to production disruptions. These failures led to improved prevention strategies, including redundant production capacity, enhanced weather monitoring systems, and emergency deployment protocols. Breeding facilities now maintain reserve production capacity that can increase output by 50% within 48 hours of emergency activation. Weather monitoring systems provide 10-day forecasts, allowing preemptive deployment schedule adjustments before storms disrupt operations. Emergency protocols enable rapid response to production failures or unexpected weather events that could create coverage gaps. Outbreak responses revealed the importance of maintaining multiple breeding facilities across different geographic regions to prevent single points of failure. When the Guatemala facility experienced equipment problems, production from Mexico and Panama facilities compensated for reduced output. The distributed production network ensures equipment failures or natural disasters affecting one facility don't compromise the entire program. However, the system requires constant coordination between facilities to maintain adequate total production for continental protection. Field monitoring systems were enhanced following outbreaks to provide earlier detection of wild fly populations. Automated monitoring stations now collect samples every 48 hours instead of weekly, reducing time between wild fly establishment and detection. DNA analysis laboratories process samples within 12 hours of collection, compared to 72-hour delays that had allowed wild populations to grow larger before detection. Mobile laboratory units can deploy to outbreak areas within 24 hours to provide on-site analysis capabilities. The constant vigilance required to maintain protective barriers becomes apparent during emergency responses. Every program aspect must function perfectly to prevent wildfly establishment, from breeding facility operations to aircraft deployment schedules to field monitoring systems. A single failure in any component can create vulnerabilities that wildflies exploit within days. Emergency responses demonstrate that maintaining protection requires treating every operational element as critical to overall program success. The biological war against screwworm flies faces the harsh reality that victory can never be permanent. Wildfly populations exist just beyond protected boundaries, ready to exploit any weakness in the sterile fly barrier. Each successful outbreak response prevents catastrophic reestablishment, but the constant threat remains. The protection system requires eternal vigilance because wild flies possess all the biological advantages that made them devastating pests before sterile fly deployment began. This invisible shield protecting civilization demands dedication that never wavers, resources that never diminish, and vigilance that never sleeps. The scale of this operation becomes clear when you consider the numbers. Right now, 100 million sterile flies protect you from flesh-eating parasites through the most ambitious biological warfare program in human history. This invisible shield operates continuously across multiple continents, preventing billions in livestock losses while most people never realize it exists. The year 2025 marks both the program's greatest triumph and its most dangerous period. Climate change is expanding screwworm habitat northward, forcing program expansion into new territories at unprecedented cost and complexity. This biological war requires eternal vigilance because wild populations wait just beyond protected boundaries, ready to exploit any weakness in humanity's sterile fly barrier.